So I'm going to tell you a little about how the yeast genetics actually came around back into the human genetics in the studies that we're doing. Thank you. And I'll be telling you about the pre-laminate pre-peptide, which we believe is a signal molecule for stem cell repair of heart and skeletal muscle. As Rick mentioned, I'm in the division of cardiology, and we were studying a family that had a disease called dilated cardiomyopathy, which is a form of inherited heart disease in which the left or both ventricles dilate in order to try and compensate for the heart not functioning correctly and in order to try and pump more blood. And while these are good short-term compensatory mechanisms, they're not good in the long term. People usually don't survive very long, requiring transplant. The family that we were studying is shown up here with the black boxes being the affected individuals. And along with having this form of heart disease, some of the patients had signs of limb-girdle muscular dystrophy, and some of the patients also had signs of Emery-Dreifuss muscular dystrophy. And the only real difference between these are which skeletal muscle groups are affected. But also what's significant is that in both of these diseases, heart disease is the major health risk for the individual. So along with the skeletal muscle problems, patients with Emery-Dreifuss muscular dystrophy or limb-girdle muscular dystrophy also develop cardiomyopathies and conduction defects and frequently require pacemakers or transplants. And so the year before we started studying this family, it was shown that families that just had Emery-Dreifuss muscular dystrophy, that in some of those families, lamin AC mutations cause the disease. And so because we had a family that has heart disease in some patients or heart disease with limb-girdle and Emery-Dreifuss muscular dystrophy, we thought that this might be sort of a carryover, and it may be due to different mutations in the same gene causing a very similar phenotype. And so we sequenced the lamin AC gene in these family members, and the affecteds and unaffecteds, and we found that all of the affected members had a truncation actually that would prevent the protein from being expressed properly. So we wanted to ask, why would a mutation in the lamin AC protein cause heart disease and muscle disease? And to start, this is a schematic of what the lamina actually is and what the lamin proteins do. So lamin proteins are so named because they basically form a laminate around the nucleus. So the lamins in the schematic form this crosshatch structure. They're filamentous proteins. They form this crosshatch structure that lies under the nuclear membranes and around the nucleus and gives it structural support, because the lipid membranes by themselves really wouldn't be very supportive, and it acts as anchoring sites and interaction sites for other proteins. It actually has a lot of described functions, although it's hard to know what the true function is in the sense of why the disease mutations are causing the disease. This is an image that we've taken with immunofluorescence microscopy. So here we've used antibodies also to stain the protein in cell. Actually, I should say this one's a GFP molecule. And so what we've done is we've taken green fluorescent protein, like the jellyfish protein that makes them glow, and hooked it onto our protein so that we can hit it with fluorescent light under the microscope and see where our protein goes. And as you can see, this is the nucleus of the cell, and it has a dark rim around the outside edge, and that indicates that the lamin protein's actually in the lamina, not floating around in the center of the nucleus. Because if it was in the center of the nucleus, you wouldn't see the intense staining around the edge, where you see a greater cross-section. And finally is this electron micrograph, and this shows the surface of the nuclear lamina, and there's a lot of globular filamentous proteins across the surface, and if you look at this nuclear pore that they've magnified here, you can see the crosshatch structure of the lamina. So as I said, we wanted to understand why mutations in the lamin gene, which is expressed in all the cells of our body and all different tissues, would cause heart disease and muscular dystrophy. And so the way we approached this was to make numerous different mutations represented by these different colored boxes here, and make the mutations all throughout the lamin AC gene, which is represented by this linear schematic. And here we have this green fluorescent protein that allows us to see the protein in cells on the end of it. And we've made mutations that cause dilated cardiomyopathy. We've also made mutations that just cause Emery-Dreifuss muscular dystrophy, as well as some that we were found to cause a lipodystrophy disease, figuring that these would be very different in what they do from the mutations that we were studying in heart disease to try and understand what the effect was that was causing heart disease. The first thing we did with these was to place them into skeletal myoblasts from mice. And so these are basically adult skeletal muscle stem cells that are responsible for muscle repair and growth of our skeletal muscles throughout life. And we placed the cells in there, and we did the fluorescence microscopy on them. And you can see in the first case that we have the wild type protein as our control. We see this nice ring around the outside edge, and the protein is localized to the nucleus. And here I'm showing you all of the mutations that cause heart disease. And as you can see, all 6 of the mutations we're studying that cause heart disease all correctly localized to the cell nucleus. However, 2 of them, the asparagine 195 lyse mutation and the ARG89 Lu mutation, cause aggregate formation, and so we don't get the filamentous lamina forming like we would expect. And so the next experiment we wanted to do was to address what happens to the proteins themselves. And so here we're looking at what's called Western blots. We've taken an antibody, we've separated the proteins by size along this gel, and then we've probed these with the antibody specific for our lamin proteins. And so the antibody actually represents a region that cross-reacts with two different splice products, and so we see the native lamin A and C in all the cells. And in the untransfected sample, we don't see this upper band, but in all the samples we put our constructs in, this upper band appears. And what that upper band is that it's the same band as lamin A, except that it's 35 kilodaltons larger because we've hooked this GFP, this green fluorescent protein, to the end of it, which has made it larger, and so it migrates more slowly on our gels. And when we looked at it, we saw that here's the wild type migration, and here's all the mutants, and we found that one of the mutations was retarded and was migrating more slowly than the rest of the cells, or the rest of the proteins we were studying. And so that small size shift we figured out was likely to be due to the fact that the protein, the lamin A protein, is expressed as what's called a pre-protein. That's expressed as a longer protein, and then it's processed, and the last 15 amino acids actually have a very specific modification made to them, and then at the very end of the processing, these last amino acids are clipped off the end, the small peptide or piece of the protein. And we thought that maybe the fact that this was retarded was because this little piece wasn't being clipped off the end of the protein. And so we used an antibody that specifically recognizes just this little piece. So if the protein had been processed that we're looking at up here, it will no longer contain this little piece and the antibody won't recognize it, whereas if it's still attached, as we believe it would be, then it would light up the fusion protein from the mutant, which is what we saw, is that the one mutant that was migrating more slowly still contained the C-terminal end, hadn't been cleaved off as it was supposed to be. We next looked at the skeletal myoblasts, the cells that are actually relevant. These are HeLa cells, which are cervical carcinoma cells. And so we looked at the muscle stem cells, and in these cells we found the same thing, except that we found that 5 of the 6 mutants smeared, and that they also didn't appear. And what we believe the smearing represents is that in those muscle cells, that this piece of the protein isn't completely cleaved off of the mutants either, that all the mutants are affecting processing in actually the relevant cell type. And when you look at the control, which is loaded at the same level, there is no smearing, as there isn't in one of the mutants didn't smear either. And so we thought we had indications that 5 of the 6 mutations causing heart disease weren't cleaving this peptide from the end of the protein. And we wanted to know why would this peptide, or the lack of its production, cause heart disease and muscular dystrophy? Because all that was really known in the literature is that this little piece, this peptide, may have some influence on targeting the lamin protein when it's first made to the nuclear envelope where it functions. But that doesn't really explain why this loss of this little peptide would potentially cause these diseases. And what we realized is that while there are no other mammalian proteins which contain this modification, which is called a farnesyl group, in which the farnesylated peptide is cleaved off after it's made, there is one other protein which is also farnesylated like lamin A, and that's the yeast A factor mating pheromone. So this is kind of where it went back to my graduate school work. And what A factor does is it's what's called a signal pheromone. It signals cells, so yeast can live as 2 haploid cells, and they live happily like that, but when they're starved or when they lose water, they want to form a diploid so they can form spores and escape the environmental conditions. And the way they do that is that the A and alpha type cells both release these peptide pheromones into the media surrounding them, and it signals the opposite cell, in this case A factor binds to a receptor on the alpha type cell, and signals it to undergo cell cycle arrest, mating specific gene expression, and then the cells undergo morphogenesis in the direction of the peptide gradient, and they fuse together to form a diploid. And realizing that, and the fact that the peptide that wasn't being made also is homologous to A factor, we realized that skeletal myoblast differentiation actually proceeds by the same pathway. And that skeletal myoblasts are kind of unique in that when they differentiate or go to form skeletal muscle fibers, we don't know what the signal is, or we hadn't known what the signal is. But when those cells get the signal, they go ahead and the same things happen. Actually, in all differentiation, the same processes happen, that you have cell cycle arrest, tissue-specific gene expression, and morphogenesis and cellular interaction. And what really set us off in this case was the fact that in the case of the skeletal myoblast, the cells actually fuse together to form multinucleate myotubes, which are the basic building blocks of muscle fibers. And so we have this signal molecule causing cell fusion, cell cycle arrest, and tissue-specific gene expression leading to this diploid. And then we have much farther in evolution in mammals that we have this system where the same process occurs, and that we have a similar signal molecule, and we thought we may have found the signal molecule that makes these stem cells differentiate. And the explanation then about why prevention of this signal being made would cause muscle disease would be because you're not signaling the muscles to repair themselves and to grow properly. So in the laboratory, we had these skeletal myoblasts, or skeletal muscle stem cells, that had been isolated from mice, but as I said, we didn't know what the signal was to get these cells to actually differentiate and form tissue. We could do it, however, by starving the cells, which was not a very efficient way of doing it. It was very slow and not very robust. In addition, when you starve the cells, they won't migrate as they would to a site of injury in vivo, that they would find that injury, migrate, and infuse in to grow the muscle fiber. But in the laboratory, we actually, because they won't migrate, if you want to make myotubes, you have to grow them to confluence so they're already touching each other before you differentiate them. And then if you starve them, the process occurs slowly. We really don't see the intermediates. We don't know what the signal is. But in the end, you wind up with these multinucleate myotubes, and they're confluent across the plate because the plate was full of cells before we actually started the experiment, and it had to be in order for them to fuse. So we took these same cells, and we actually looked up the concentration at which, the lowest concentration at which yeast A factor was active, and we tried that same concentration of our peptide that was chemically synthesized on these myoblast cells by just putting it into the media. And so the cells weren't confluent, and we weren't serum starving them to differentiate them. And when we put that signal into the complete media, I hope this shows up, but here are the untreated cells, and within 6 hours, our cells, an intermediate filament protein localized to the nucleus, but more significantly, you can see that the cells have migrated out of these open areas represented by the arrows and have fused together already in just 6 hours with the area where the triangles are. And when we let these plates go for out to 3 days, what we saw is that we got sheet-like, this is a triangular myotube, angled and curved myotubes for the first time. And as you can see, there's no confluence and there's no other cells next to these, that the cells migrated in together and they fused together to form myotubes. And so this experiment represented the first time that myoblasts were induced to differentiate in complete media, the first time that they differentiation occurred without having cell confluence, the first time they migrated and fused in the same assay, and the first time that they've been induced to differentiate in minutes and hours versus days. And it's also the first time I'm aware of that we've generated curved and angled and sheet-like myotubes, as opposed to the normal linear myotubes that would normally form. In addition to this, because we were giving them what we believe is the native signal, we saw what we believe are intermediates in the differentiation process, or this tissue formation process, that have never been seen before. And so while the lamina, as I've shown you on the previous slides, are all this continuous, kind of ubiquitous, homogeneous film that covers the nucleus, when you treat the cells with this prelaminate peptide and then look at lamina expression, the lamina changes. The laminas become this very exact structural architecture that, and the blue signal here is DNA, that surrounds and organizes the DNA. And as the cell goes farther along in the process of differentiation, the lamina forms, I don't know if you can see them, but tetrahedrons off the surface, and seeds, which doesn't show up at all here. There's a red signal. I don't know if we can shut off the lights for a second. Thank you. No, if that helps. But yeah, so you can see that there's actually, the cells kind of come together, and it still doesn't show up here, but there's a very crosshatched red signal indicating that the lamin formed at the nucleus is seeding the cytoskeleton, sort of like an ice crystal would seed a snowflake. And then here we have a cell that we believe is much farther along in the differentiation process, and you can see that now the lamina has completely covered up all the blue DNA signal, and that we have much more filamentous structure as the lamina forms the cytoskeleton and seeds the nucleus. And this is another image, and these are at 6 hours after treatment, so this is less than half a day. And here we can see that the cells are responding to each other and are sensing where each other are and sending out filaments, so they actually begin to attach together so that they can form these myofibers. And so this cell here is polarized in the direction of where the other cells are, and it sent out this filament, which actually looks like it's curling out in a corkscrew type pattern as it tries to find the gradient, and then attaches itself to these cells. And then these cells have gone farther along, and we have multiple filaments forming between cells as it's going to fill in and form tissue. And these cells down here are much farther along, and you can see again that there's so much of the cytoskeletal architecture that's formed at the nucleus that you again can't even see the DNA, that's completely coating the nucleus as it's growing out and forming the tissue. So what I've just shown you is that when we treat skeletal myoblasts with this peptide that we've identified, that they fuse together and form myotubes, and that we see intermediates that hadn't been seen before. So skeletal myoblasts form myotubes and fibers because basically skeletal muscle acts like rope to actuate the levers in our body, which actually occur at our joints. In contrast to this, closely related cardiac myoblasts form triangular heart cells, and these cells come together not by fusing together, but by excreting what's called extracellular matrix, or basically stitching, that stitches all these cells together into sheets. And the idea of this is that, as opposed to contracting a rope to pull on levers in our body, we want the sheets of tissue in our heart, when they contract, to decrease their surface area such that the volume of the heart decreases and we eject blood from the compartments. So the model for cardiac myoblast differentiation are called rat H9C2 cells, and these cells are cardiac myoblasts isolated from embryonic rat aorta. There's a problem with these cells, though, in that, again, we don't know what the signal is in the laboratory to make them differentiate or form tissue, and so we serum starve them to get them to differentiate. And while these cells were isolated from embryonic rat heart, and when you serum starve them, they make heart-specific proteins as opposed to muscle-specific proteins, showing their origin is heart. what you get morphologically is nothing that looks like triangular heart cells or tissue, but instead you get linear myotubes, just like you do with skeletal muscle. And so it was not really clear, and people didn't really know, is this an artifact from the experiment in the isolation of these cells? Do they really represent cardiac stem cells? It wasn't really clear, but it was the closest thing people had to work with to making heart proteins, and this is what's been studied in the laboratory. So we basically treated these cells at the same concentration with the prelaminate peptide, and what we saw is instead of them fusing like they do to form myotubes, that within 6 hours they excrete extensive amounts of extracellular matrix, or the stitching material that holds the tissue together. And so if you look at this nucleus here in blue, you can see that this cell has secreted all of this extracellular matrix, all of this red signal, the stitching material, in just 6 hours. And similarly, this nucleus here is filling in the space around it with extracellular matrix as well in just 6 hours after peptide treatment. You actually see this within minutes before your eyes under the microscope. And what was really shocking is that when we let these things go out to 2 days, for the first time, these cells formed sheets of striated tissue. And so you can see this tissue sheet running across the slide here horizontally, and you can see there's a nucleus caught in the edge of the tissue sheet where it's peeling away from the slide right here, and all the space between the cells is completely filled in with extracellular matrix, or the stitching. You can also see that there are some binucleate cells you see in real heart tissue, and I don't know if you'll show up, I guess you can kind of see it here, and is that what we also saw is that there were striations running within the cells and between cells, showing that there's higher order tissue organization. And so as far as I'm aware, this is the first time that cardiac stem cells have been induced to form tissue without actually layering them on some sort of a matrix that already looks like tissue, that these cells actually have all the information apparently to begin the stages of forming cardiac tissue by themselves. And so that was a pretty exciting experiment. And then in the end, what we believe this shows is that by treating both when we treat skeletal myoblasts, they very robustly, as measured by the number of nuclei within each myotube, fuse together to form myotubes, doing things that they'd never done before in serum starvation. And when we give the cardiac myoblasts the very same signal under the same conditions, that they instead form sheets of striated tissue for the first time, indicating that the peptide is not some sort of a chemical modifier that's allowing cells to fuse together to binding to the surface, but it's actually signaling two very different processes in two very closely related cells. So we believe the applications for this technology are that the prelaminate peptide may be used to generate tissue for implantation, for drug discovery, for testing for toxicity rather than going into humans, and that the peptide itself provides an exciting new therapeutic avenue to treat heart disease and muscular dystrophy, because right now where everyone is trying to isolate stem cells or take stem cells and differentiate them towards the site that they want and reapply them to the body, we believe that this technology provides a method potentially to be able to give a person that's sick a signal molecule by itself as an injection, as you would with insulin, another peptide, and induce their own stem cells to repair the damaged tissue. Thank you. Are there any questions? Thank you.
Is there an element of targeting required when you inject the...
I think that's one of our largest concerns, is that when you picture these things actually, this molecule actually being able to direct how tissue forms, that there's a very good chance that it may be anchored onto the extracellular matrix itself in order to make the cells differentiate from the tissue in the way that they should. And so at this point, it's not completely clear if we can just give somebody like a vitamin shot, a low dose of this, and we'd induce all the stem cells in their body to potentially repair things or go specifically into the heart, or if we're going to have to anchor it and target it in other ways directly to the tissues that we want affected.
Do you have any in vivo models for this? Or?
We're going to animal testing, yes, yes. There's a number of diseases that are caused by inhibition of prelaminate processing, and it seems that there's a dose-dependent effect on how many different types of tissues. It actually seems to expand beyond skeletal muscle and heart. And Hutchinson-Gilford progeria, for example, the premature aging disease, is also caused by inhibition of this peptide production. And so based on the literature, it seems that you get a dose-dependent effect, that the more you inhibit peptide production, the more tissues are affected, and the more severely and earlier that the disease pathology seem to show up. But we have to go into that sort of plan as well. Thank you.